Well, first, Rena, thank you very much for having me here. So, senescent cells, a very interesting topic. So, senescence is a cell fate. It's what happens when cells acquire a lot of damage. This can be a variety of damage. This could be genotoxic stress, it could be mechanical stress, or it could actually be mutations that can lead to cancer. And the cell says, something's wrong. I'm going to stop growing. So the cell shuts down, it exits the growth stage, and it starts to release factors that tells the immune system, I'm damaged, it's time to come clear me. So it makes a lot of inflammatory factors, which allows the immune system to come clear those cells. The problem is as we age, our immune system doesn't clear those cells as effectively. So they start to accumulate. So you have these cells which aren't growing that are very inflammatory. And as you get more and more of them, that inflammation drives tissue destruction and contributes to aging. Right. So senolytic drugs, it's actually a term that was coined in collaboration with a group at the Mayo Clinic. So these are drugs that specifically will kill a senescent cell. So when cells undergo this cell fate to become senescent, they change a lot of parameters in that cell that now make it susceptible to certain classes of drugs. For example, a senescent cell in some ways is a precancerous cell because I said mutations that can give rise to cancer lead the cells to start to grow and they undergo this state of cellular senescence. So anti-cancer drugs in many cases actually will kill a senescent cell. And so these are drugs that will kill a senescent cell, a tumor cell, but not a normal cell, causing them very quickly to lyse and disappear from the body. It's a great question. It's always funny how your career path takes different directions, but about 10, 12 years ago, I started to work on senescence. I wasn't really sure how important it was for aging at the time, but it was one of the processes that we know happens with age. But what makes it very attractive is that it is druggable. It is possible to develop drugs which will eliminate these cells, termed Senolytics. And we could develop ways in culture to screen for these drugs, which will kill senescent cells. So unlike other types of things that go wrong with aging, we thought that senescence was targetable. And then shortly thereafter, there was several landmark papers that came out of labs at the Mayo Clinic with collaborators of ours that really demonstrated that if you got rid of these cells genetically in transgenic mice, so these were engineered mice, that allowed you to clear these senescent cells with age, that these mice were healthier for longer and they lived a little bit longer, suggesting that you could develop drugs like Senolytics that eradicate them or reduce the level of these cells, that it would improve health span and potentially extend lifespan. Well, so first, I just like to go on record saying we're not trying to make people live longer. So the goal of my research and many people in the field is to keep us healthier for longer. And if the byproduct of being healthier for longer is living longer, so be it. But we want to live in a very healthy manner. So senescent cells in general probably drive aging. So if you clear them, it's going to slow the aging process. But having said that, it's been shown by other groups, and we have similar data, that eliminating senescent cells can improve the quality of the endogenous stem cells. So each tissue in your body has a stem cell that can allow for repopulation of your liver, your kidney, muscle, other tissues, maybe even the brain, although that may be in question. So if you reduce the senescent cell burden, the quality of these stem cells improve, which may allow you to repair damaged tissue, maybe potentially live a little bit longer. But right now, we're really hopeful to keep us healthier for longer. And if you live a few years longer, healthier, so be it. We'll take that. There are hallmarks of aging which have been identified. There are now 12 hallmarks. These are things that can go wrong with aging. And if you exacerbate them, you age faster. And if you target on them, you age slower. But they're all linked. So if you affect one of these hallmarks, the other 11 hallmarks seem to improve. So NAD metabolism is indirectly linked to senescence. So if you treat cells with an NAD precursor, NR, NMN, or others on the market, you can improve mitochondrial function. And if you improve mitochondrial function, it actually reduces senescence and inflammation. And if you drive cells into senescence, there's mitochondrial dysfunction and the NAD levels drop in your body. So they're all linked. So I think if you're taking an NAD supplement, an NAD precursor supplement, it probably reduces your senescent cell burden to some extent. And if you reduce your senescent cell burden, it probably increases the level of your endogenous NAD levels. So they're all linked. I think that NAD precursors in animal models have been shown to be very effective at extending health span. The data in humans is still not as solid. And I have to say, for many supplements on the market, there's actually very little evidence that they work. Some might, but very little evidence that they work. So you have to be very careful about what supplements you take and really have scientific data to really document that there is a positive effect. The other thing we don't know is the dose of these drugs to take. What's the dose of an NAD precursor and how often should you take it? And these are things that require clinical trials to determine. So self-medicating with a supplement sometimes doesn't lead to a benefit. NAD can improve mitochondrial function, which probably reduces the percent of senescent cells in your body.
Okay.
So it also increases the activity of enzymes that probably modulate senescence. So it does have an overall effect on your senescent cell burden. In theory, it should increase your energy level, so you should be more active. You should feel less tired. Whether it helps your sleep, I don't know, but I would imagine if you're more active and feeling energetic, you're probably going to sleep better. But again, for humans, I think the data is still lacking. The clinical trials are ongoing. There's positive data with several of these NAD precursors, but more studies are needed. But in mouse models, it does increase energy, increases activity in different models of aging and disease. So senescent cells have certain markers or things that the senescent cell has in it, which are different than normal cells. But it's not different than all types of normal cells. So there are certain things which maybe are predominantly found in senescent cells that maybe are only infrequently found in normal cells. So we measure these markers, and you do a before and after treatment with a senolytic, or as you age and you see the level of these markers. Now, does that say that the cells have disappeared or that the markers have disappeared? So the cells could still be there, just the markers have disappeared. So really showing loss of cells due to death caused by a senolytic is extremely difficult because it's very indirect. All we can say is that markers of senescence are lower in somebody taking it, or in a model of aging that's taking the senolytic. What's the best target for senolytics? That may depend in part on the senolytic that's being used. But there are a number of diseases or even disorders or conditions where this could be applied. So, for example, there are clinical trials starting to try to get rid of senescent cells in an old liver that's being used for transplant. So it's been suggested that getting rid of senescent cells in an old liver, old kidney prior to transplant will allow that organ to work better. It's possible that recovery after surgery, if you're older, if you have a bone fracture or if you tore a muscle, it may be the clearing these bad senescent cells will allow for better repair after that procedure or after damage to a tissue. So it doesn't have to necessarily even be a disease. It can improve our resilience. We have published that the mortality, at least in animal models, of a COVID-like infection is reduced by reducing your senescent cell burden because it improves resilience. Now, having said all that, everybody wants to pick a disease where the outcome is not good for the aging individual. So many companies, many laboratories are focused on things like pulmonary fibrosis. So idiopathic pulmonary fibrosis, if you're diagnosed with it, has a death sentence within 5 years. So the thought is if you could extend lifespan by clearing the senescent cells that accumulate in your lung, that would be a big step forward, or potentially liver fibrosis. There are clinical trials ongoing with Alzheimer's disease, suggesting if you clear senescent cells in the brain, it can help with cognition and memory. We've been doing studies in Parkinson's disease. So a whole spectrum of disorders or conditions. Another example is that there's evidence of increased senescence in Down's patients. So, and there are genetic diseases of children that are allowed to accumulation of senescent cells, like Hutchinson-Gilford disease, Cockayne syndrome. So it may be possible to treat even genetic diseases in children with senolytics. So which ones are the best target? I think it's going to take clinical studies to sort that out. Probably sometimes. The example I'm thinking of, there's a drug that's been shown to be a senolytic. It's called navitoclax. It's a drug being used for cancer treatment that targets a family of proteins that block apoptosis of tumor cells. And some senescent cells increase the levels of this protein that prevents them from dying. And so this drug is very effective at killing tumor cells, senescent cells, but it turns out cells like platelets in your body also express this protein. So the drug actually will kill platelets, which causes complications in humans. So I think what we're going to discover is that there is no one target in the senescent cell that's highly specific only for that senescent cell. It may be predominantly found in the senescent cell, but there may be a cell type somewhere in your body where that senolytic may have an adverse effect. However, having said that, the advantage of senolytics is that they can be given intermittently. So the thought is if you give them every 2 weeks or every month to kill your senescent cells, it targets the senescent cells, although if it kills some good cells, they can come back and still be there, but the senescent cells have been cleared for a period of time because they're more effectively targeted. So the thought is intermittent treatment will make senolytic drugs safer than if you had to take the drug daily, such as you would have to do for cancer therapy. And so this is kind of a controversial question in the aging space. There are people who think that we can extend lifespan 150 years, 200 years, or indefinitely. My view is a little bit more restrained than that. There was a very nice study published by a collaborator of ours, Jan Fike at Albert Einstein, who looked over all the aging records of every population in the world, and he came up with a very nice study that says the maximal lifespan is 115 years. And there's been a few examples of people living 116, 117, but that in general is the maximal lifespan. I think senolytics are not going to extend it past 115. The goal is to allow more people to make it to 100 or 105 and to be healthier. So that we're not in nursing homes, we're not going to the emergency room every 6 months with a different disease, different ailment. And I think even the mouse models where we really can control the number of senescent cells which have been killed, we're not seeing huge differences in lifespan. We might see a 10% increase, 15%. So I don't think we're going to see much with senolytics. If we did, if you saw a few extra years, that's great. But again, the key is to keep us healthier for 3, 5, 7 years longer than we currently are. If you live longer, you live in healthier because you can just extend the period of time that you're in a nursing home. So the numbers, just give you some numbers about healthy aging. So if you're 65, like I am, I'm 66, the chances are I'm going to have 2 comorbidities. And when you're 70 or 75, the chances are you're going to have 3 or 4 comorbidities. And that means that you're not having just one disease, you're having multiple diseases. You go to your doctor, he gives you a drug for this disease, gives you a drug for that disease. You go back, a drug for another disease. There's no indication those drugs actually don't cancel each other's out or cause adverse effects. So they treat each disease, disease at the time. So what we want is kind of a way of actually slowing down the onset of these comorbidities and reducing the severity. And the best way we think to do that is to target aging itself, to try to reduce the number of comorbidities. Can you eliminate them all? Can you reduce the severity of them all? I think for each of us, we're all different. I think each of us has different comorbidities. So it's going to be really different for each one of us. And so some of us with senolytics might live a little bit longer, some a little bit less, but hopefully we live healthier. I think the field has moved forward with senolytics much faster than I would have expected. So in 2015, Jim Kirkland, Tamar Ciconia, Laura Niederhofer, and myself described in an aging cell paper the first senolytic combination of dasatinib and quercetin In 2018, we described the ability of a natural product, Vaccinate, to actually have senolytic activity. And these compounds now are in dozens of clinical trials in the United States, clinical trials around the world, and being offered by clinics that have been set up in different countries around the world. And what we know is that these drugs actually can reduce somewhat, depends on the disease, the age, or the condition, but can reduce senescence. And so, knowing that, we're now hopeful we're going to be able to show in the next couple of years that actually improves human health, at least some conditions. And that will be an interesting question. Does it really slow aging? Does it treat certain disorders, certain conditions? So my guess is, depending if it's a natural product or if it's something you need a prescription for, like the Dasatinib or Navitoclax type drug, um, the natural products will be probably 4 or 5 years before everybody's taking them for aging. If it's a prescription drug, it'll probably have to be 5 to 10 years. It'll take a while to do those phase 2 and phase 3 trials to really document the therapeutic effect. I think this is more historically, there's been very poor science performed in the aging space, but the potential to make a lot of money. So there are a number of drugs which are marketed as having the ability to improve your appearance or potentially able to slow aging. Drugs like something called Provence that's advertised on late night TV, that's a jellyfish extract that improves memory. But the data showing these actually work is very, very slim. In fact, many cases, there's no data. So physicians who know something about science and biology are skeptical because there's very limited data. I think the field has changed in the last 10 or 15 years. Quality science has now entered the ability to do testing in model organisms, whether it's in worms, which is a nice model system, lives 2 weeks, or fruit flies that live 2 months, or mice that live 2 years, or non-human primates that live 25 years. That sort of data is now telling us, yes, these compounds can have effect on health span, have effects on lifespan at this dose, these combinations. So there's now much more data behind it that says, yes, these compounds work. What we now need is evidence that they work in humans, because not everything that works in a mouse or a fruit fly is going to work in people. And so that's the next big step. And when we see positive, well-controlled, well-designed clinical trials that show benefits, physicians and other people in the aging field will say, yes, this works. Well, it's an interesting question. I think it really depends upon the culture. So, for example, in Minnesota, a lot of the indigenous populations, believe that with age comes wisdom and respect for elders. And they're very hesitant to take treatments that might extend lifespan because aging is a natural process. And they actually view Alzheimer's as part of that natural process. And I think there's some other cultures that have similar views. And so I think it really depends if you value the insights of your elders or if you think it's time to move on for younger ideas and younger input. But again, the point that we're trying to emphasize is we're not trying to make people live longer. We want to make them healthier where they can contribute to society more than if they were infirmed in the nursing home. And there's also then the economic aspect of this, which we haven't talked about. And the cost, the economic burden of taking care of 25 or 30% of your population who are over 65, who don't work, who are getting Social Security, at least maybe for a while longer, we'll see, but Social Security and health benefits, that cost has been projected in 20 or 30 years to be the current cost of the defense budget. So there's economic reasons to keep people healthier, and I think actually societal reasons because of the knowledge and wisdom that the older people have. I don't want people to live up to be 150, 200, 250. I think then that issue becomes more of a problem. You're right. If they're the ones dominating that society or dominating the views, like an Elon Musk. Interesting question. So I think you could look at both the contributions of some of these billionaires to aging research in both a positive and negative light. Clearly, it's brought a lot of money to the field. It's brought a lot of interest to the field, and it's helped move the field forward. I think the downside is sometimes it's provided maybe a little bit of overhype, maybe more than it should. And then this desire to develop something to live longer, not necessarily healthier, but to live longer. And some of the approaches are not affordable to the average person. So there's the view that these people are designing effective therapies to allow them and other rich people in Silicon Valley and other places to live longer and healthier, whereas the average person cannot. So as we go forward, I think it's important for all of us in the field to realize we're trying to develop approaches to keep people healthier for longer for everybody. And that's important to consider with doing clinical trials. We shouldn't do clinical trials in older white males. We should think about doing this both in males and females because they do age differently. Different ethnic backgrounds have different drivers of aging. Incidence of diseases are higher in Hispanic populations and African American populations and Caucasian populations. So we have to think about doing this in an equitable way that allows everybody to have an opportunity to live healthier and potentially a little bit longer. So I think the billionaire contribution has been very important, but I think they have to understand this is to allow us to develop ways to keep everybody healthier, not just an expensive treatments such as plasma infusion or bone marrow transplants or genetic engineering that only allow those with money to live healthier and longer. So currently, the field's evolving. There seems to be more pillars and hallmarks added every couple of years, but there are currently 12 hallmarks. Very recent paper published by leaders in the field. And they're really broken down into three types of hallmarks. They're the primary hallmarks, which are the drivers of aging. Then there are those that respond to those hallmarks, which kind of can mediate the effects on aging. And then there's the third layer that further mediate the effects. So one of the drivers we think of aging, a few other drivers are DNA damage, or kind of genomic instability, changes in the epigenetic regulation of your DNA, which means they're not mutations in the DNA code, but modifications of that code that allow it to be read differently. So this changes with age. Stability of proteins. These are things that do enzymatic activities in your body. How they're made and how they're broken down changes. These are primary drivers. And then the secondary drivers are really broken down into three, which are senescence, mitochondrial dysfunction, and nutrient sensing. And then these lead to things like chronic inflammation and change in your gut microbiome, your body's microbiome. So what's interesting is the three hallmarks of all those hallmarks, which are targeted for therapeutics primarily, are mitochondrial function, which many things that reduce the levels of reactive oxygen species, improve mitochondrial function, so antioxidants. Things that regulate nutrient sensing, and these are rapamycin and rapalogs, which are in clinical trials and shown to allow mice and other model organisms to live longer. And then cellular senescence, which is what we're focused on, which is, like I said, a state, a cell fate that we think is easily druggable. And those are the three areas which are the three hallmarks which are being targeted by aging researchers and companies focused on aging. And I should add, though, that the primary hallmarks, like how to repair your DNA, change your epigenome, improve protein stability, those may be tougher things to target. And that's obvious by the fact that there's no clinical trials trying to target these primary drivers or primary hallmarks of aging. It's really the secondary effects, which are the more druggable ones. So there are many levels of how AI can help the research. So just focusing on senescence as an example, I'm part, as many of us at Minnesota, are part of a consortium that tried to identify, characterize, and spatially map senescent cells with healthy human aging. So what that means is we're using all this cutting-edge technology to identify, look at the metabolome, the lipidome, the transcriptome of senescent cells that arise in different tissues and different parts of the body over time with age. And this is huge amounts of data, which a normal person can't digest. and sort through and try to figure out where the senescent cells are in a given tissue. And that's where AI can help. So trying to identify a senescent cell, characterize a senescent cell, and then take that information and say, okay, what are potential drug targets? We identified that in the portal vein of a liver of a 75-year-old person with liver fibrosis, there are these types of senescent cells that are accumulating. What potential targets can we identify in that population of senescent cells that we can develop drugs against? And can you help model the type of drug we should be synthesizing that might work as a senolytic? So drug development, identification, characterization, this is where AI can play a huge role. And that's a problem with the field, is that as we learn more about senescent cells, we realize that they release into the environment many of the same factors that immune cells do in responding to an infection. So we're trying to identify markers, and we have a few, but we'd like to identify more that distinguishes a senescent cell from an inflammatory cell. I think a simple answer would be usually if you have an inflammatory response against a bacterial pathogen or viral pathogen, that inflammation is transient. It comes up and goes away. We view senescent cells as leading to chronic inflammation that is maintained. So if you see this higher level of inflammatory factors that's maintained, that we would view as more likely due to a senescent cell or different types of senescent cells. So I think time is an issue, and then there may be markers, some of which we're starting to identify and more which we have to, that will separate an inflammatory senescent cell from the inflammatory immune cell. Because that's going to be important when we try to look at the role of senescent cells in inflammation with aging as compared to the role of inflammation in clearing a pathogen. I think the answer is yes to all of them. So it depends on the senolytic. We talked about senolytics as drugs. These are being small molecules, but there are other approaches that are being developed. So there are antibodies, which may target proteins on the cell surface that are preferentially there on the senescent cell. There are therapies developed for cancer called CAR T therapies, where you engineer a T cell so it targets like a tumor cell. This is being done to make a T cell so it targets senescent cells. And there are different types of small molecules which are being developed. So how do you target a senescent cell in the liver? You have to find a drug that actually first will engage the right pathways in that type of senescent cell in the liver. You have to make sure that drug actually preferentially goes to the liver. So if you have a drug trying to target senescent cells in the brain and it doesn't cross the blood-brain barrier and doesn't get to the cell, it's of no value. So it depends on your target. If it's a liver, kidney, muscle, pancreas, wherever. And so you can engineer small molecules so that they target certain tissues. You can engineer molecules so they might be preferentially binding to and taken up by senescent cells. You can use biologics. Like I said, biologic therapies, antibodies, CAR T cells, other approaches along those lines also are showing effectiveness. So it really is going to depend on the senolytic, the disease, and the the senescent cell target. Right. So question in the field, and I think we still haven't answered this. And there's some people to say it's independent of the type of senescent cell. We just want to reduce the overall senescent cell burden by 50%. It doesn't matter what it is. We just want to reduce that level. There are other people say that there are specific types of senescent cells that we need to specifically target. But I think either way, if you have to target a specific type or two, or if it's just generally reducing all senescence, it's going to be a cocktail. I don't think there's any one target in all senescent cells that will allow for that cell to be specifically killed. So I think we will have a combination which may involve biologics and small molecules, or all small molecules, or potentially all biologics, that we'll be taking. The question is, do you take them all at the same time, or is it that you take them, all of them together every 6 months, some every month, some every 3 months, whatever? I think the timing is going to be very important, and the concentration is going to be very important. So eventually it'll be a cocktail. But what it's targeted against, is it just all senescent cells or a subtype of senescent cells, is still yet to be determined.
Yeah.
And I guess the other thing I can mention, and we'll have to see how this plays out, but there have been some very nice studies showing that you can vaccinate against senescent cells. So this is relying on the fact that certain types of senescent cells express proteins on their surface, which are, I won't say 100% specific, but preferentially on those senescent cells. And it's shown you can vaccinate and then their senescent cell burden stays lower. The concern, obviously, is if you vaccinate against the self-protein that's expressed on normal cells, that eventually could cause problems. So that's going to have a lot of safety testing. But it could be that you get immunized when you're 20 against a senescent cell antigen or two, and that keeps your senescent cell burden low and you're healthier and you live potentially a little bit longer. But I think that's the concept with small molecules, and people are trying to design them to make them stealth molecules. They're only activated to kill the cell once it's taken up by a senescent cell. So, I mean, there are a lot of really interesting approaches using nanoparticles to deliver toxic compounds to senescent cells. Lots of different approaches. What's eventually going to make it, out there for the public to take? I think simple right now is better. So a simple natural product that's effective at reducing senescent cells, that's going to be the first thing on the market. Eventually, there will be far more sophisticated senolytic approaches.
Yeah.
I think the one limitation, though, and it's something we haven't touched on, is everybody ages at a different rate. So if you don't have senescent cells, there's no reason to take a senolytic. And some people may have senescent cells occurring at 40, 50, 60 years of age. Some people might not have senescent cells till they're older. There's aging differently. They have a different genetic makeup. So what we need are ways to identify who has the highest burden of senescent cells and should be taking senolytics more aggressively. And we see this in mice. If you treat a middle-aged mouse that has no senescent cells, there seems to be no benefit. If they're starting to develop senescent cells, you clear them, there appears to be a benefit. So biomarkers to identify that population is going to be very, very important. Well, so there are numerous clinical trials underway to test senolytics for different age-related diseases. But one of the endpoints for those is actually taking a variety of bodily fluids and tissues that are accessible and measuring everything we can measure. So part of the clinical trials testing senolytics are also going to help identify markers that show responses to senolytics, which may correlate with your senescent cell burden. So this is all going to go hand in hand, but identifying biomarkers, and that's true not only of senescence, it's mitochondrial dysfunction, stem cell function, amount of DNA damage. You can go through a long list of all these hallmarks of aging, but trying to quantitate those to determine if you should be taking a senolytic or this other person should be taking a stem cell factor, or this other person should be taking something that improves mitochondrial function. Eventually all of those things go wrong, so you should take all of them. But who should take what when they're 40 or 50 is what we need to determine. You just can't run out there and everybody take a senolytic when you don't have senescent cells. Yes, we're very, obviously we're very heterogeneous. We're much more heterogeneous in our genetic makeup than an inbred mouse strain where we do most of our experiments. So just to talk about some of the complexities. So one of the things that we study are centenarians. And so the question is, why does a centenarian live to be a centenarian and not develop any diseases till they're in their 80s or 90s, or even till they're in their hundreds? And there's a lot of evidence that says it's due to their genetic makeup because it runs in families. So we, working with a group at Albert Einstein, at Columbia University, have been sequencing genomes of centenarians. And we've identified rare variants in different cohorts of centenarians that are linked to longevity. And now we're trying to develop drugs that mimic those rare variants to see if they will now allow those of us who are not centenarians to live healthier. But what's interesting is many of the variants we're identifying in centenarians seem to reduce the rate of senescence onset, at least in assays we can do in cell culture. So we think that one of the reasons why centenarians live longer is they have a variety of genetic changes that reduce their senescent cell burden, probably because they can repair damage. They can deal with the damage, they can deal with the stress more efficiently. So it may be the clearing senescent cells is going to actually be another way of mimicking what we see in centenarians that have reduced levels of senescence and a better response to stress.